Okay, good evening, all.
Good evening, sir.
Right. Today we have started our lesson and we have just discussed about some of the factors related to the living beings. So today also we are discussing about our first lesson, so which is the living world, the wonderful living world. So we learned that we have already learned. So the living world has three spheres basically. So these spheres facilitating the living organisms. So what are these three spheres, if you can remember? So basically you have the biosphere with atmosphere, lithosphere and hydrosphere. Vayugolaya, Jalagolaya saha Bhugolaya. So we have three components in our biosphere. The living organisms lives there. So basically the environments, then we discuss different environments. Maybe our indoor home environment, classroom environment, maybe ocean or forest. Different environments are there. In these different environments, we can see different objects. Some are living and some are not living. So that also we discuss in different environment. So either it can be artificial, man-made, or it can be a natural environment. So doesn't matter whether it's a natural or artificial, the living organisms live both environments. In natural environments, also artificial environments, living organisms are there. We learn about that. Then we learn about different ecosystems. Actually, these systems are, some are natural, some are man-made, and some are mixed. It has natural and man-made both components. We learn about them as well. And then we learn about one of the main features of this living organism, which is growth. So every living organism has this feature, growth. Non-living organisms also, some of the non-living organisms, they got this feature, but mainly the living organisms, has the feature of growth. The growth කියන එක ජීවීන්ගේ ලක්ෂණ. Growth is feature of living organisms. But you can see the pencil, it's not drawing, the buildings are not drawing, but you can see a puppy, it has drawn a dog. So it's common to us and animal. And so we saw how it happens, how a baby come to the world, we discuss about that. And also we discuss about these seed and their maybe the germination process. Yes, we learn about germination process. Yes, and also some other drought related processes related to plants and animals. And I think, yeah, this is germination. We discuss about that. And then we discuss about some other important factors related to the living organism.
Learning how the plants grow. So, in today's episode, let us answer an evergreen question. What is seed germination? Zoom in.
Okay. Germination also related to the growth. The growth එකටම තමයි germination. Germination කියන්නේ seed එකක් පොඩි අපි මොන හරි බීජයක් දැම්මම ඒක ඉපදෙන එක, ප්‍රරෝහණය වෙන එක. So that is called germination. That is also related to the growth. So then, not just that, there are some more features related to these organisms. Nutrition. Nutrition is essential for the growth. We can take nutrition from food, basically, and sometimes through drinks, but that also can be taken under food or the beverages. Or the another important thing is reproduction. So either sexual or non-sexual reproduction is there for these plants and as well as the animals. Reproduce means create in the next generation, generate in the next generation. So the babies. Organizing is also a feature of these organisms. They can organize, they can work together, they can interact with the environment and also interact with the ecosystem. So that is organizing. Evolution. I discussed that evolution means we believe that the human evolution happened from monkeys or gorillas, whoever, whatever. So anyway, the evolution happens for long period, for thousands of years, this evolution happened, and now we have Homo sapiens sapiens, the current format of us, the human. Not only that, even the animals. You know the dinosaurs. Earlier days, in films, I think you have seen the dinosaurs. They are no more because they are not here. They have, so they are organisms related to them, and their reproduction system is totally stopped. Now we cannot see any dinosaur in this world, but still we can see some fossils or ruins related to them, because the evolution is happening, but after some, due to some of these natural catastrophes or some scenarios, these species can vanish. Adaptation, extinctability, coordination, metabolism, respiration, movements are also some other related features related to growth. Okay, today what we are going to learn? Today we are going to learn more about these things. How we can categorize our surrounding or our environment. So let's list the things. So can you tell me some organisms around you? I think you have seen plants, animals, microorganisms, and human also, us. So those are organisms. Anything other than this? Any other organisms which come to your mind? You can tell. Okay, so what's around you, Pute? Can you tell me the things around you? I think you can see your desk, computer, right? Maybe your pen, pencil, books. Those are not non-living. Can you tell me few more? What you can see around you today? Yes? Come on. Yes, please tell me. Yes?
Things only organisms.
Yeah, anything. So non-living organism. Okay, tell me an organism.
I
Organism.
have a little clock.
Sorry? Frog. Is it?
Clock. Clock.
Clock.
Yes.
Oh,
It belongs to non-living things.
okay. That is non-living. Yes, that is non-living. Any living organism around you? Maybe your pet belongs to animals, right? Maybe plants. Okay, these are the things. So what are microorganisms, Pute? Right, maybe waterfall. These are also waterfall, mud, all are. So there are a lot of non-living organisms, non-living things. Those are not organisms, non-living things. Okay, so you can write down this today. So organisms, please write down this table. So also the question, list living and non-living things in our environment. So please write down this question, list living and non-living things in our environment. Living and non-living things in our environment. Then write down, then draw this table. Finish writing. Tell me if you're finished. Finish or not yet? Okay, write it quickly with it. I have a small question. So what are microorganisms? Right? Microorganisms. Do you know microorganisms? Or have you seen? No. Okay. Microorganisms microorganisms are very tiny, small organisms which you cannot see in your naked eye. Using your naked eye, you can't see them. So those are called microorganisms. Here, these are not microorganisms. You can see these things. These are clearly visible to you. Elephant, trees, lion. You can see it's clearly visible. A flower like lotus. But these microorganisms, you can't observe them in using your naked eye. Naked eye means original your eye. You can't see them. Those are around our environment, everywhere. In the air, in water, in food, even inside our body, or in our body surface, even in our skin. These are everywhere. Millions of microorganisms. So think about corona or COVID-19 virus. It is a microorganism. You can't see, but sometimes it's not falling under organisms. But samaharu කියනවා සමහරට that viruses are not organisms. Viruses are not living beings. It has living and non-living features both. Okay. So, as I said, virus is can be categorized as a living or non-living, because virus has living features, non-living features both. When it comes to our body, when it moves to our body, it start reproduction. So we can call it is a feature of, it is a living feature. Reproducing is a living feature. Reproducing can යාගේ copies හදනවා. When it comes to our body, අපේ ශරීරයට ආවට පස්සේ it can create copies. යා copies හදාගෙන යාගේ පිටපත් හදනවා. එහෙම තමයි අර කොරෝනා ටිකක් අපි කියන්නේ වැඩි වෙනවා කියලා. So what happens if let's say a certain concentration of virus is actually infected or infected to our body or respiratory system, then it comes to it will be move into our cells and start reproducing themselves, copies. So it will increase the number of copies and increase the effect. It will use our reproduction system or the cell reproduction system to reproduce. Our cell, අපේ තියෙනනේ අපේ සෛලත් හැදෙනවනේ. ඒ ක්‍රමය අරගෙන එයාගේ සෛල හදනවා. So that is bit odd, right? It's like using someone else's machine and getting copies. Let's say you go to a shop and getting photocopies. The photocopy machine is not belongs to you, it's belongs to the shop. But you can still go and take copies. Viruses are doing same. Virus come as a single copy. The photocopy machine is our cell. Our cells are the photocopy machines. So it will photocopy or copy and spread. So that is what happens. So viruses sometimes not considered as a microorganism or living organism. But bacteria, yes. Protozoa, algae, fungi, and archaea also falling into the microorganism. Have you seen one of these? Anyone? No. Oh yes. Fungi, I think you have seen fungi, right?
Yes.
Fungi, it's visible because it's micro, but when it grows, so isolated fungi, maybe you cannot see, you cannot observe that using your naked eye. But when it grows as a group, then you can see. When you spread as a group, then so let me show one example. Here, let's go to, okay, let's say you can do this as a practical in your home. Take a piece of bread. Okay, take a piece of bread and small part, right? So this is then keep it. Let's say bread and fungi, right? So you can keep this. Here you can see cheese. So this can be done for cheese also, right? Bread, or you can see.
Observe the bread mold fungus in the laboratory. You can grow and observe the bread mold fungus in the laboratory.
Okay, Budhe. So see this video now, right? Api make a bala. So you can do this as a practical. Yedrat karla balanna puluwa. How? Take a piece of bread. Then
To do so, obtain a piece of damp, stale bread and leave it exposed to the atmosphere for a few days.
but you have to make it wet, right? Then if you like put some water into this, it will increase the growth. It will expand it. That growth will be increased. You can accelerate it, right? By putting water. If you just put the piece of bread, it will take some more time. But if you put some water, then within 1 or 2 days, you can observe this. Not even one, not within 1 day, you can observe. කරලා බලන්න ඊළඟ දවසේ විසි කරන්න චූටි කෑල්ලක් ගන්න ගොඩක් ගන්න එපා පරාදෙන්න. Just take a piece of bread, put some water into it and keep it exposed to the environment. Then after some time you can see
You will notice a furry growth appearing on the bread.
okay, you can see this is accelerated, right? This is slowly happening, but this is accelerated. You can see some patches. These are signs of fungi. These are signs of fungi. But the thing is, individually isolated fungi, you will not be able to see them. Your naked eye. තනි එක්කෙනාව බලන්න බෑ. Right? You will not be able to see individuals. But you can see the organization, or you can see the group or the growth cluster. මේ ඔක්කොම draw වෙද්දී අපිට පේනවා. But as like individual, you cannot see that.
Leaving the bread exposed to the atmosphere increased the likelihood of fungal spores from the air settling on the bread, growing on it. Let's have a closer look at this fungus. Transfer a small bit of the growth onto a microscope slide and observe.
So this is very important equipment. So what is this, Pute? Have you seen this?
Yes.
What is that?
A microscope.
Yes, it's a compound light microscope. That is the full name, right? It's a microscope, that is true. It is compound. Compound means it has more than 2 lenses. It's compound and it's using light. Compound light microscope. So a microscope can be used to observe. මොකද අපිට සාමාන්‍ය ඇහෙන් පේන්නේ නැහැනේ. We cannot look at this using our naked eye, no. Then we can use a microscope. When you use a microscope, it will enlarge this. Then you can see the fungi.
Mucor is a multicellular fungus. The fungus is organized into mycelia, which is made from thread-like structures called hyphae. Most hyphae contain many nuclei and are said to be multinucleated.
Okay, this part we learned in higher grades, right? Now you know how to observe that, no? Now you know how to do this experiment. You can try this next day and you can even send pictures to our group. Okay, that indicates that you are doing the practical. But I know that you, do you have microscope in your home? Anyone?
No, sir, not yet.
No, right. So what you can do, Pute, you can create your own. So I'll tell you how to do that. You can create your own microscope. So for low cost, if you like, you can try to create that. I'll show you next, right? Okay. Anyway, this now you saw this fungi, but there are some more bacteria. I think, are they helpful or harmful? What do you think? Are they helpful or harmful?
Some are helpful, some are harmful.
Some are helpful, some are harmful. Actually, which are helpful? Can you name helpful one? Yes? Any helpful fungi or any helpful micro? Binuti, I observed some dirty water from our school compound microscope, and there were years, there was some, yes, true. So we'll do that practical also, right? We'll learn that practical also. Some are helpful. So, you know, some fungus, actually we used to eat them. You know the mushrooms, right? So those are also belongs to category of fungi. Some kind of fungi we can eat. We can use as a, not just us, but some animals. Some animals they eat, right? They eat that. And so it's useful. And some bacteria are living inside our body, help us digest in some food even. So those are healthy. And some bacteria are there in our skin, help us protect our skin from bad microorganisms. So some good microorganisms are there in our body. So some are inside, some are outside. So අපේ ශරීරයේ ඇතුලේ හෝ පිටත ඇතුලෙත් ඉන්නවා එළියෙත් ඉන්නවා මයික්‍රෝऑर्गेनिज्म ඉන්නවා උඩ ඉන්නවා අපේ ශරීරය අපේ හැමේ ඉන්නවා අපේ ඇඟ ඇතුලේ ඉන්නවා ඉතින් අපිට මෙයාලා අයින් කරන්න බෑ සම්පූර්ණයෙන්. So these microorganisms, right, these microorganisms, so these microorganisms are helping us. එයාලා අපිට උදව් කරනවා, right? Sometimes these microorganisms help us fight against bad microorganisms. නරකයත් එක්ක fight කරන්න, to fight with bad microorganisms, these microorganisms are helping. Right? These microorganisms are helping, right? These microorganisms are helping to protect our body or protect our organs from bad microorganisms. Again, naraka kattiyegen protect wenna udav karනවා. Microorganisms are helping us protect from some bad microorganisms. And even fungi, like some microorganisms, animals take these microorganisms as food. Even algae. So some like sea animals, or some like some animals are there in these. So we learn about this water-based animals. So they are using this algae as food. Fungi, these things, and bacteria. Actually, bacteria. So do you like yogurt and all? Or these food-based products, yogurt, food-based products, or these milk-based products, not food-based, sorry, milk-based products, cheese, yogurt. Do you like them? No, you don't like. No, you don't like, okay. So even to produce them, some microorganisms are used. The curd can be produced using some bacteria, this lactobacillus. And even yogurt industry, curd industry, some microorganisms are used too. And yeast is used in creating some, the flour-based products. Yeast can be used. So it's used for cooking, even producing some food, and also for cooking, and also as a food we can use. So some are really helpful. Some microorganisms are helping us, but some are bad. They're not helping. So viruses basically kind of bad microorganism, right? Basically, it gives a lot of troubles. Okay, so these are some enlarged or microscopic pictures which are taken from microscope, a light microscope. Chlamydomonas, these names, don't need to remember them, but just showing. This is Chlamydomonas. This is amoeba. Chlamydomonas, amoeba, paramecium, euglena. These are some microorganisms. So let's see how to observe them. So let's learn how to observe. Observe karanne kiyala. So let's see how to observe. Okay, so as I said, you can use the compound microscope. And here you can see some categories of bacteria. Coccus, Bacillus, Vibrio, right? These are basically based on the shape of the bacteria. Different bacteria shapes. So you can see them. But don't worry, no need to remember these things. Just remember that different bacteria are in different shape. That's enough. No need to remember the shapes and shape names. And you can use the compound light microscope to observe it. And as I promised you that I will show you guide video or DIY video which help you to create a microscope at home. This is very easy. You can also try. So let me show the video first. Okay. So can you identify what they have used? So it's very simple, right? They have used plastic, these plastic pipes. So these are there, right? I think you have seen these plastic pipes are there. Then this is objective lens. This is objective lens. So if you want, you can buy objective lens for very less price from even Daras. You can order from Daras. So it's very small amount. So no need to pay a big cost. And not just that, you need another pipe. And they are sticking this together. And this is another one. You can see there is 9-inch. This here 9 inches eyepiece tube. So it's just putting, this is another lens, eyepiece lens. It's also very small lens, right? That will also not cost much. And then 2 lenses. So since there are 2 lenses, we call that a compound microscope. So I think you have seen this one. Have you seen this hand lens? Have you seen this hand lens, Putin? I think you have used that.
Yes, sir.
That is why. So can we see these things using hand lens? Microorganism, can we? It's difficult to think, right? It's difficult to see microorganisms using hand lens. So that's why we are using compound lens. Tiny lens it's only single lens, so it's very, very difficult to see the microorganisms using this hand lens, and that is why that is why we are using compound lens, compound microscope. Compound microscope has 2 lenses. The previously installed 1 lens, you can see here they have installed 1 lens, and there will be another lens after that. You can see the second lens. So because of these 2 lenses, the magnification factor is achieved. After that, they mount this together, and you can see, right, this is small, like you can see small cover, and there is a hole. There's a reason why this hole today. This hole lets the light travel. This is a light, you can see small LED light. This hole let the light travel to the lens through this. When you set the light, you can see the light is going to the lens through this hole. Actually, this hole you can keep a glass. So this glass is called slide. So what is the name of glass? It's called slide, right? S-L-I-D-E, slide, right? This glass is called slide. And then you can put your specimen. Specimen means what you are going to look. Then you can put your specimen here. Actually, they are putting small leaf. And after that, observing through the lens. And you can see the view, right? Here you can see the view. Actually, this is testing, right? To focus, they use this. To focus, actually he has used this leaf. Then you can see a drop of water is put. So as who said that? Binuti said that. Binuti, are you there? Binuti, are you there? I think your name is renamed something, right? Okay. Then, so after putting this drop of water, they are observing. Then they can see small worms-like organisms, right? Small. Your name is changed. The Yashoda means I think your mother's name, right? I think this is what you have done, Binuti, right? Is it? But you have used a proper microscope. Microscope use. So this is homemade microscope, but still possible to see these microorganisms. So let's give a try. Just give a try. Maybe you can create a homemade microscope, which you can keep into your science exhibition next time. Right? So in your school, when there are science exhibitions, you can show this as your work. So it's not bad to do DIY. Do it yourself is always better. You can buy a microscope. I know there are microscope in shop, so it will not cost much. But still, if you can create your own one, then you know the technology even. It's good opportunity to you to experience the technology. Okay, do that alone. Just watch the video again and you can do that. Okay, so here's what Binuti has done. So I'm going to explain that. You can put some dry grass or maybe right dry grass or some dry leaves to this water and keep for few days, maybe 2 or 3 days. You can keep it. Then in this water there will be lot of microorganisms after 3 or 4 days. So what they are doing, they are just putting this grass into the water and keep it for some days. Actually, here grass and water, you can just see. They keep this for some days. A question. Yes, dry hay. Dry hay is better. So that is the practical they are in your book, Pute. Better to put dry hay, right? These are grasses, like, but dry hay is also good. Hiduru neda, single dry hay. You can put that. Then here, this is called specimen, Pute. You just take a drop of water, and this is your slide, and you have to cover this slide using that is here if they are not covering, but it's better to cover this slide using a cover slip. So there's a small thin glass which is called cover slip. You can cover it using a cover slip, but they are not covering that. They're just looking at the microscope. Okay, you can see here the movement is another feature of a living being. You can see the moving. You can see the size. This is 1 mm ruler. Ooh. So very small, tiny organisms. You can see amoeba. Here, protozoa. So I'll name them. I'll tell Paramecium and all. You can see them. Different organisms. When you see, you will get this. And you can see they are eating, swimming, reproducing. Here creating copies, reproducing, producing a new cell. Here you can see bacteria. Bacteria are this small, smaller than other organisms. And the shape of bacteria is also there. But this cell is a bit larger. The other cell. These are bacteria, all bacteria. Looks like worms, right? But here see this large cell. That is not bacteria. It's a protozoa. But so basically there are more bacteria and viruses, but you will not see those using the light microscope. You need electron microscope to see them, because light microscope, the magnification level is not enough to see these viruses and all. This is just one drop of water, right? Okay. Then shall we write the experiment, Puti? So as Binuti said, we are using the Hay, we use Hay infusion. So we call that Hay infusion. So this is how you officially, or you write a scientific experiment, right? In scientific experiment, first you have to write the heading. Experiment is identifying microorganisms in Hay infusion. So that is the heading. So this method you have to remember, right? So this is the very first experiment that you are going to do in grade 6. Right? Experiment is identifying microorganisms in Hay infusion. Okay. Then materials. Materials is what you need. Definitely you need Hay infusion. No? Hay infusion, how to create Hay infusion with it? You can put dry hay. You can put dry hay into water and keep it for some days, maybe 2 or 3 days. You can keep it exposed to the environment. Then there are a lot of bacteria, viruses, and what you call this protozoan, algae. Those are there in the environment. So they will come, they will attract to this environment, the Hay infusion, and start growing and start reproducing their organism. Basically, they start reproducing because they have water, they get food. So when there are food, water, and the media, which is good to them, so they start reproducing. That's why after 3 days, after 2 or 3 days, you can see a lot of thousands of bacteria, these protozoans, and all. That's why you can see them in this drop of water. Okay, Pute, so let's remember these shapes, right? Because this is not, this will not be asked in your examination, but just remember the shapes of this. This is very important, right? So these amoeba, chlamydomonas, euglena, paramecium, these are protozoa, right? And bacteria, you have seen, right? Bacteria is this, right? Bacteria is protozoa. Basically, they have both features, right? Protozoa, actually, they have both features. Means they have features of animal and plants. So that is in between. So protozoa is a different group. Algae, so algae, you have seen this in surfaces of water, in tanks, maybe in your well water, in tanks. So there is a green layer. This green layer is algae, right? So basically, non-moving water, there are a lot of algae. If water is moving, then algae will not be there, but there will be some few, but not much. But if it is not moving, a tank-like thing, then it will spread very soon. That is green color, easy to recognize. Yes?
Sir, is there algae in seawater?
Seawater, there are some algae. Yes, there are. Red color one. Have you seen this? This is green color one. No, this red. Red. Ocean. So basically, it's come here. Have you seen? So sometimes in some, this is kind of algae, right? You can see it's in the ocean. I have not seen this in Sri Lanka much, but I have seen in some videos, right? I have seen in some videos. This is a type of algae in ocean. So it looks red color. It looks like blood, right? This is due to algae. Sorry?
It looks like pink milk.
Yes. It looks like. So it is in seawater, right? So there are some oceans affected with this red algae. And this is also red algae, but you can see it's a different color. So this is red algae again in ocean. So it's there. So even so, these are there in even very extreme environments. Even in volcanoes, even in deep ocean, there are microorganisms. Can you believe that? They are in some extreme environments. Extreme environments කියලා කියන්නේ සාමාන්‍යයෙන් ජීවීන්ට ඉන්න අමාරුයි. Volcano is really, really hot. But there are some microorganisms. In deep ocean, no light even. In deep ocean, light will not be there. It's very dark. But there are some microorganisms. Right? ඒ වගේ තැන්වලත් ඉන්නවා. So ocean is nothing to them, right? In the ocean කියන්නේ යාලට ගාණක්ම නැහැ මේ මුහුද කියන එක. Because even deep sea volcanoes, glaciers, glaciers are like ice cubes. Ice cubes in this Atlantic area or north and south. So even in glaciers, there are some microorganisms living. So very cool. Volcanoes, very hot, but there are. So they will definitely be in the ocean. Right. Bacteria, protozoans. So just remember the shape and the name. Protozoa is a different category. Bacteria is a different category. Then archaea is a different category. Virus is a different category. Fungi is also a different category, and algae. So these are the categories. Right? Just remember the names. Okay, shall we write down this? I think better write down, right? So did you start writing the that experiment part? Have you started? ඔයාලා පටන් ගත්තද පුතේ ඕගනිස් අර එක්ස්පෙරිමන්ට් එක ලියන්න පටන් ගත්තද? Or not? Yes or no? If not, please write this microorganism and their names, right? Don't draw this. Drawing is not necessary. I'll give you the slides, right? As a PDF, I'll send you the slides, but just write type of microorganisms. So just add small part, type of microorganisms, types of microorganisms. And then please write bacteria, archaea, protozoa, virus, algae, and fungi. Today it is very, very important to write the experiment with these steps. So please remember these steps. So this is your very first experiment, and you are going to do some more experiments in future, actually in the science. So you have to learn, and not just the teacher can give you some knowledge, but in science you have to behave as scientists. Scientists are doing researches. Scientists are experimenting, doing on researches. So it's very, very important. Scientists are researching, doing on researches. So be like scientists. Become a scientist, a junior scientist, and start exploring, start experimenting. And this is your very, very first lesson. Very first experiment, experiment. So in this, first of all, you have to write down the experiment name, and then the materials. Hay infusion, you can just put some hay, piduru, into water and keep it for few days, 2-3 days enough. Then the compound light microscope is needed, and glass slides. Why glass slides? That is where you are going to put drop of water. Before you watch, you have to put drop of water to the glass slide and cover using a cover slip. Right? First of all, so what is the method? The third thing. The first thing is experiment. The second thing is materials that you need. The third thing is the methodology or method. The method is you just take water sample, and that is your hair infusion. Place a drop of water into the glass slide and cover using the cover slip. Then place it in the on the stage, and stage of your compound microscope. And then stage is the place where you put these slides, right? I'll show you. Observe the slides using low power microscope, low power of that microscope. So please write down this. I'll show you it. First of all, please write down. Is so do we need to that step is not yes. Do we need to clean the microscope glass slide and other parts, because then there can be harmful microorganisms? Yes, true. It's very true. Correct. So actually that part I didn't add it to here. So I assume that the Microsoft is, slides and all these are already clean. But it is a good practice to clean them before you start a new experiment. Why? Not because of the harmful microorganisms, because you cannot find where these microorganisms came. Let's see, you keep the slide as it is without cleaning. The mega karla may slide let's think that you keep this slide as it is. Slide without changing. Then what? Then after doing that, then you try to right after that, then you try to put this into a new experiment. Then you can't find out where these microorganisms came. It can be from previous experiment. Then our assumptions are wrong. That's very, very good point. We can add that to our note. Yes. So very good point. Okay. So what is that? So I'll put double asterisk and write there. So that point is very important. Make sure all the slides and cover slips are well cleaned before new experiment. Before new experiment. Oh, sorry, it's my mistake. Before new experiment. Make sure all slides and cover slips are well cleaned before new experiment. That is required. Very good. Very good point. Okay, Pute. Then, right? Okay. So with that, Pute, we have come to end of our today's lesson. So we discussed a lot related to these microorganisms, and we need to learn the broad classification of these living things again as a summary. So what are these living organisms? Actually, what are these categories? So let's look into that. Right? Let's look into that. What are these categories? Okay. So the last slide says that. So last slide for today, right? Next day we will discuss the other part. So classification of living things. So basically, it belongs to 3 domains. There are 3 domains. So the domain 1 is bacteria, right? The kingdom 1 or domain 1 is the kingdom bacteria or domain bacteria. Actually 3 domains. And archaea is the second domain. So you have archaea under that. So archaea is type of bacteria basically living in extreme conditions. It looks like archaeobacteria කියලා. You can call archaeobacteria. That is also type of kind, looks like a bacteria, but a different organism again. So then you have eukarya. Eukarya domain, it has a lot of sub kingdoms. So kingdom Protista, kingdom Plantae, kingdom Fungi, and kingdom Animalia. So these are the subdomains related to these. Okay, let's write down this quickly. Classification of living things. No need to draw the picture. No need to draw the picture, right? So just write domain bacteria, domain archaea, domain eukarya. So those are the 3 domains. Under domain bacteria, just give me a second. Okay, under domain bacteria, this kingdom Eubacteria is domain archaea, kingdom archaea. And domain eukarya, they are Protista, Plantae, Fungi, and Animalia. Protista, which is like amoeba-like thing. Plantae, the plants, trees. Fungi, so basically the fungi. And then the Animalia. Those are the 4 types under domain eukarya. Okay. I hope we have discussed the related things. Finish writing? Okay, Pute. So let's wind up with this for today. Next day, I'll be discussing some more features related to this living organism. This is a lengthy lesson, even though we have to discuss much more about these topics in the next day. Then good night. See you all next week.
Good night, sir.
Okay.
Sweet dreams.